Good morning, everybody. I'm very happy to see you and delighted to present Carole this morning. Carole is going to talk about the continuation of a research that I started many years ago, and she kind of transferred the research from France to Colorado, you will see. And I presented a couple of years ago the presentation to the entire group. Carole, I had the fortune, like that Carole came to me one year and a half ago, roughly, and she asked me, "Do you have something to give to me as a research?" And I said, "Yes, of course, I do have the perfect stuff for you." And she has been wonderful. She has, carry on the research independently, although Carole is an undergrad, but she has demonstrated independence and so on. And I would like to point out that with this research, Carole won the first prize in our Jackson School Symposium, the last one in February, before the lockdown. So I would like to let the stage to Carole.
Okay, so I'm going to share my screen. All right, can everyone see that? Or Connie, let me know.
You're good.
Okay, great. So today I'm going to be talking about the biotic influence on speleothem morphology. So first, what is a speleothem? And it is a calcium carbonate cave deposition, and these are the 6 most common that we see in caves. And then there are some really weird ones called halectites, and these grow sideways against gravity in all sorts of directions, and their formation hasn't been completely explained yet. So the motivation with this kind of research is that speleothems are part of the geologic record. This means they can last billions of years and can give us clues about the past. Caves are generally considered inhospitable because obviously they don't have any light. They have very little organic carbon, sometimes little water, it depends on the cave, and they can have heavy metals in the soils. So even though they're considered inhospitable, there's still an enormous diversity of microbes, which means they're using some sort of unconventional energy source, and understanding this energy source could give us some information about how life can survive that we don't know yet. Microorganisms precipitating speleothems is a rare process as of right now of what we know. So also it would be a rare discovery. And there's a medical impact about this kind of research because understanding extremophiles, because caves are considered an extreme environment, and any microbe that would be living in this environment would be considered an extremophile. This research would help with antibiotic and antibiotic resistance, as Buehler's has wrote a paper about. The last thing is that caves have UV shelter from UV rays, because UV rays destroy carbon. So if there's one spot that we would find life, for example, on other planets, it would probably be in caves, as they would be protected from UV rays. So the first site that we're going to be talking about is a cave called Asparage in France. And Nicola is going to talk about that one.
Okay, I will steal 5 minutes only here, because people probably know already what has been done there. So after 2-3 years of work, in 2015, we kind of show that the speleothems that you see here in this cave in France, where their formation was controlled by life. And we found a series of microbes or a community of microbes living nearby this concretion and on the concretion themselves. So the discovery, please next slide, was done in the south of France. You see here a geological map in an area where there are Carboniferous and Permian series, sedimentary series, and the rocks are maybe marbles, and there are marbles and schist, so like shales, kind of rich of heavy metals rocks. So please next slide. So the blue gallery was discovered in 1994, which is this area here. The blue gallery is called like that because the concretion in that area are blue, despite this one which are white. And the question of a friend of mine, who was also a speleologist, was why are they blue? And then he called me in this cave, and then as soon we approached the cave, me and other speleologists, we kind of realized that there was more to understand than simply the color of such speleothems. So please, the next slide. The interesting thing is that such bouquet of speleothems, they develop only in a very restricted part of the cave, only a few hundred meters, while the cave is several kilometers. And they grow on a substrate, which is, again, very rich in heavy metals. And so on. So please, next slide. Yeah, so we found a series of morphologies that kind of convinced us that the formation of such concretion should have been driven by biotic influence, so by life, right? And to this, we have, for example, very long branches of this concretion that they grow one towards the other and meeting the center of the room that might suggest quorum sensing, which is the ability of some microbes of sensing other communities of microbes. We have also large bites, like here on the B part of this figure, right? Or concretion that grows against gravity and so on. So please, next slide. So the question, once we saw such concretion, the question was clear. We sparked immediately the question in our mind: are these more similar to a concretion like the one that Carole showing at the beginning of the presentation, or more like a coral? And is there life controlling the formation? And then please, next slide. And then immediately we realized that this part of the cave, in association with this concretion, there was a series of microbial communities that you can see here in panel C and B, and there was, and these were they were tightly associated with such concretion. Please, next slide. So we also found the evidence of life on the concretion themselves, like filaments. We also find very weird stuff like insects that were trapped inside the crystalline structure of the concretion and so on. So this kind of convinced us further that life was probably having a role in the morphology of such concretions. Please, next slide. So I'm skipping here a lot of analysis, including CT scan and SEM that Carole will show later. But at the end of the day, we arrived to demonstrate that the microbial community present on the concretion can indeed form calcium carbonate. So we try to play a bit with agar plates inoculated with calcium, and this formed very interesting structures after a few weeks. The same exact white dots. Please, next slide. So the structure that you can see here, these dots surrounded by the dashed line there, those are nodules of calcite that were formed inside the agar plate. And these were formed in a few weeks. So we kind of demonstrate here that the microbial community that is associated with those concretion can indeed deposit calcium carbonate. And then I let to Carole to finish.
So finding another location that also has the same morphologies as were found in Asparage would strengthen the hypothesis that these morphologies could be created biotically. So there was a second site in a cave called Breezeway in Colorado. So I'm including some pictures so you can see the similarities and the fact that these are growing against gravity and sideways in weird ways. And you see this encrustation up here. So a little bit about the geologic setting. I put a red dot. It's a entire park, actually, where the cave is located. So this is a map of the entire park, and this is just where the cave is. So there are limestone units and shale units and a lot of different rock units in this area, which, as Nicola mentioned earlier, then you have heavy metals that we'll talk about in a second. And the comparison in between Asparage and Breezeway, these are just field photos, but just looking at the general morphology, you can see a lot of similarities between the two. So given the similar morphology, the question is the genesis the same? So to answer this, first we did field observations. So we looked at field pictures and compared them to Asparage, and then we gathered samples from the floor. So they were gathered from the floor instead of off of the actual stems because this cave is protected and we'd like to go back and get better samples, but we performed analysis on those that were already fallen off. Then we took micro CT scans. This is a non-destructive imaging technique so that we could look at the inner morphology of the samples and create 3D models. They were taken by Eric Goldfarb at the Chevron Digital Petrophysics Laboratory. Then we did X-ray diffraction. So I hand ground some samples and then placed them on a little plate. And then Thomas Etzel helped me with the analysis in Phil Orlandini's lab. And then we did SEM analysis. So this took some prep. We freeze dried in Tam Shanahan's lab the samples, and then we gold coated samples in Phil Orlandini's lab several times. We chose gold coat over a carbon coat because we were going to do EDX to search for carbon. So we chose gold over that. The SEM done was at SC Ukar's lab at BEG, and it was done with Sarah Elliott. So same as Asparage, there's several kilometers of cave, but the only spot where we see these helictites is just in this area. So it's very localized. In both Asparage and France, the same order when you're walking into the cave of morphologies is found. First, you start seeing these white dots on the walls and the floor and the ceiling of the cave. And then you start finding this encrustation of calcium carbonate, and then the helictites growing off of that encrustation. And this is in Asparage, and this one's in Breezeway, and the exact same order is found. These white dots were found to be microbial communities in Asparage, and they were only found near Speleothems, and the same thing is true for Breezeway. So they're only in the cave, they're only found near Speleothems. So as Nicola was just talking about, we see these bights and bridges, and it seems that these Speleothems are growing towards each other and in strange directions, so this could suggest quorum sensing, as Nicola described earlier. In both cases, we also see the helictites growing off of a paleosoil with high concentration of iron and aluminum. This means that microbes that live in this environment, it's another evidence that they're extremophiles because it'd be hard to live on soil with heavy metals. So we see two distinct morphologies in both caves. We see tubular, which is made of calcite, and it has this hollow center. And then we see acicular, which has just this micrometric tube in the middle, which helps explain the abiotic formation because of capillary pressures. And this morphology has already been explained abiotically. And then we have a hybrid, which is both tubular and acicular. We see calcite on the outside and then aragonite on the inside, and the formation is still yet unknown of hybrid, though the acicular has been explained to be abiotic, and we hypothesize that tubular is formed biotically. So this is a field picture of a hybrid morphology. So if we look closer, we have the tubular right up here, and then the acicular right down here. So to kind of follow along, the tubular looks like this in a hand sample, and the acicular would look like this in a hand sample. So with XRD, we found that the acicular samples are made of aragonite and the tubular are made of calcite. And though not included here, we did do carbon oxygen isotope analysis. And when we did that, I had to get little powder samples of both acicular and tubular samples. And I found that the tubular were much easier to break you had to be very delicate to not destroy the whole sample. While with the acicular samples, I really had to put some muscle with the drill to get any powder off. So it was an interesting comparison between the hardnesses of the two. So this is comparing tubular versus acicular. You can see in the CT scans that micrometric tube, and those aragonite needles are pointing up right there. And then the tubular, we have the crystals pointing inwards with a relatively hollow middle tube. This is comparing tubular samples from Asparage and Breezeway. In both cases, we have somewhat of a hollow inner with calcite crystals growing towards the center. And then if we look closer up here, in both cases, we see this low density layer in CT scans that's sort of covering the crystal with this layer of air. So I programmed 3D models to say wherever the density was of obviously what was a crystal, I would say make it yellow. And then what was that low density, I said make it red. So as you can see, there's this low density layer covering a lot of the crystal. This is comparing Asparage and Breezeway acicular samples. In both cases, we have that micrometric tube and that characteristic aragonite needles sticking up. This is a 3D model made using micro CT scans. This is a sample from Breezeway. So keeping it together, we have tubular sample, hand sample, and then this is the CT scan of that tubular sample. Same goes for the acicular sample. These are the SEM results of the tubular sample. So that low density layer that we saw in CT scans, we actually figured out was this layer up here, and there is that layer of air. It's not a solid crystal as this one is right here. And we saw this on all of the tubular SEM scans that we did. And we also saw these filaments that were connecting these calcite flakes, this layering right here, to each other and to the larger crystal. We know that this coating right here is also calcite because we did EDX on both. This is another picture of that coating. And so this is a different sample, and you can still see that there's that coating. This is seen from a little bit farther away. I'm talking about this one. Now, acicular samples, we mostly saw these bare, undisturbed aragonite needles as well. So they're very different from the tubular samples. So bringing it together again, tubular all the way from large scale to very, very small scale, and same for acicular. Now about hybrid morphologies. This is a scan taken from an Asparage sample. So we can see that's tubular up here, relatively hollow, and then it's acicular down here. This is a 3D model of the same scan. And same goes for this Breezeway sample. It goes from tubular down to acicular. So the hypothesis that we have is that those white dots, those microbial communities, are inhabiting that wall and the ceiling, and then they get onto these branches of Speleothems that are originally abiotic. They were created abiotically and were all aragonite and acicular. And then when inhabited, they would transform the aragonite into calcite somehow, and they would deposit then this calcite right here. And so the theory is the hybrid morphology just means that the microbes haven't transformed the entire branch of Speleothem yet. So we interpret this layering of calcite on top of the larger crystal as a construction site. So microbes would be in the middle of creating more crystal. And these filaments that are connecting the flakes, it's a little bit hard to see in this image, though whenever you're looking at the actual SEM scan while it's being done, you can really tell that this shape is sort of goopy, while these calcite flakes surrounding it are very structured, angular. So you can tell there's a very different morphology in between the two. So these filaments are probably EPS, which is extracellular polymeric substances, which is just a fancy way of saying snot. It's leftovers from microbes that might have been there before. So as I said, most acicular samples had no EPS, and then we found some with these weird structures that were only found near disrupted crystals. So as you can see, so these are all acicular samples right here, and you can see the bare needles right here, and then a really disturbed needle right here that we zoomed in and found this guy. And then whenever we found this disrupted crystal, we found this one, and over here is the beginning of this one. So these suggest that this sample was in the middle of being colonized and then transformed. So when comparing construction versus dissolution, we see this one appears that there's already a solid crystal that is being grown on top of, versus the acicular sample, which looks more as though there's these, kind of looks like cheese, like there's holes being put into the sample. So it appears more to be dissolution, though this is just a hypothesis. So overall, we compare Asparage versus Breezeway and see a lot of similarities between the morphologies and the white dots that we found. And this strengthens that the original hypothesis that was given from Asparage, that these speleothems could be biologically created, or their creation would be biologically mediated. And given that there's caves present on Mars and other planetary bodies, and these speleothems could be used in the geologic record as they could last billions of years, then we suggest a potential way to search for past or present evidence of life, as we could just go into a cave and then see this. If we prove our hypothesis, we could see the morphology and say at some point there must have been life here to create this specific kind of morphology. So more questions we have is, why would microbes even form these structures? And could we find these on other planets? Along with many other questions. So I'd like to thank my team members for helping me out, faculty and staff who helped me with all my analysis, and the Cave of the Winds for access to the cave, and all the guides for finding the specific helictite site. And are there any questions?
Thank you so much. You showed such really beautiful images. I was very, very impressed. It was really, really gorgeous. And I have a lot of questions, and I'm sure other people have too, because it's just so exciting. Maybe people can raise their hands once they have a question, and I will take the advantage of being the host and asking one first. And so you said that you find these different structures localized in those caves.
Yeah.
And I was wondering what these localities might have with one another in common, that there might be a little bit more hospitable than maybe other places.
So that, I mean, Nicola can correct me if I'm wrong, but I'm pretty sure we don't exactly know why. We just noticed that both in Asparage and Breezeway, there's only a very small area. So it's more of like, hey, this is the same in both places. That's kind of weird.
Yeah, I think that's really interesting. Okay, please, Rowan, go ahead. You were the first one to raise your hand.
I can chime in here. It might be the availability of heavy metals, but we can discuss this later. Yeah.
That actually segues pretty well into my question. So I think, Carole, I think we talked about, first of all, great talk. I think we talked about this a bit one time. So when we're talking about microbially induced carbonates, have you heard of the La Brea tar pits issue?
No, I have not.
So just like the La Brea tar pits, you can get organisms like mammoths and stuff stuck in the tar pits, even though they're not forming the tar pits. And so this has been a big question with microbial carbonates is, are the microbes forming the carbonates? Are the processes that they're doing as part of their respiration influencing the carbonates, or are they just getting trapped in the carbonates? So I don't expect you to have an answer for this, because that's always something that everybody struggles with. But do you maybe have some thoughts on how we might be able to tell whether these are microbially precipitated, just influenced by the fact that microbialites are around and doing whatever kind of metabolic pathway that they're doing? versus just getting trapped. I also have an issue with your caves being inhospitable. There's a lot of life in caves.
So it's possible that there just happens to be life there. This is just a hypothesis. We'd have to lab test. I mean, as Nicola did, he created the carbonates and he did all of that. We'd have to do more analysis, of course, on these speleothems and get better samples. We know that aragonite has a higher free energy than calcite, so the hypothesis is that at somehow transforming from aragonite into calcite, that energy is how the microbes would be getting energy. Though you're right, I mean, this is just a hypothesis.
Well, there's been a lot of work, I'll send you a paper. There's been a lot of work in like marine carbonate precipitation looking at different microbial metabolic pathways and what those do to alkalinity. And that could certainly change, you know, the favorability of calcite to aragonite or vice versa. So it's certainly an interesting idea and something that you might, you know, depending on what the funding is like for this, Nicola, you know, maybe thinking about doing something like 16S or figuring out sort of the community composition there might help us figure out might help you figure out the microbial metabolic pathways.
You can say us. We have done that for Asparage.
Okay, cool.
I think,
so who's there?
Oh yeah. It's there. It's all in GenBank. But I think she has. Carole has some slides about that, so we can discuss over those slides maybe. Yeah, it's a big question. It's a big question, Rowan. It's true. We don't know if the metabolic processes of these microbes simply generate calcite and the microbes escape from the entombment, right? Or if they control the morphology. Now, if you look at the quorum sensing, this might suggest that they control the precipitation, because I don't know. Yeah, if you look at this image here, right? What are the odds, right, that those two concretion grow exactly one against the other and meet in the center of the room? The probability are pretty slim, and it happens several times in that room. So there's for sure this question: if they control the precipitation of the mineral, or if they just, you know, if just something that happens and they kind of escape it, and therefore, yeah, that's the final morphology. Yeah, here is the DNA sequencing. Yeah, maybe. Okay.
I mean, there's nothing wrong with being microbially mediated instead of or influenced instead of mediated. I mean, it's still an interesting biotic signal.
No, I agree. But yeah, it's the. I mean, it's interesting thing is that, and Carole can chime in here. More we look in these things, more question we have. That's the interesting thing.
That's true.
Right.
But that's good news.
Yeah,
that's good news.
So,
okay.
Can somebody explain the DNA sequencing that's on the screen? Just like what's our takeaway from what we see here?
Yeah. So these are, well, I don't know, Carole, if you want to discuss this, if you study enough the paper.
No, you can talk about this. I'm good.
Yeah. The takeaway is that first of all, the community is formed by a series of different microbes. They are all bacteria. We never find fungus or fungi, right? We didn't find archaea, which was also very weird. There is another slide here that maybe can help better to understand what. Well, this is the GenBank. So if you go back, there should be another slide with triangles. We don't. Okay, no, don't worry. We don't have that slide. So if you go back to the sequencing, yeah. Now I have another slide somewhere in some presentation that shows that as you move from the wall to the tip of the concretion, the community gets smaller and smaller. So you have much more bacteria on the walls and in those white dots, and you have very few bacteria instead on the tip. I'm talking about, you know, diversity here, right? And the interesting thing is that, for example, in the white dots, we find bacteria that are specialized in dealing with copper. Okay, and they're found, for example, in old copper mines and so on. So I don't know if this responds to your question, Sean.
No, that's great. And then these actinobacteria, are they? It seems like there's quite a bit there.
There is There is also one species, roughly 11% of this biome that is not really. It was unknown. However, to create a new species, you need to have more than that. You need to have more analysis than that. So there is the potential of finding new kind of bacteria in that cave, in Asparage. These are Asparage. We are missing the Breezeway. Now we would like to do exactly the same thing for Breezeway.
Sure. Yeah,
and
cool.
Carole has been talking with biologists. Maybe you want to talk about.
Yes. Yeah, we're trying to do more, get better samples and do more biology analysis in Breezeway. But yeah, he's right. This is Asparage.
And the Myxococcal is there, which is this 2.4% over here. That is known to have quorum sensing, for example.
Thank you. So next is Yiwa. I'm sorry if I mispronounced.
Okay. Very nice talk. This is Jihua. So I have two small questions. One, you did mention that when you prepare the sample, it seems that there's a big difference between the hardness of the minerals formed biotically and abiotically. I'm curious why it is that. And the second question is that have you analyzed the chemical composition of the abiotically formed carbonate and the biotically formed carbonate? Maybe there's some difference between the partitioning of the transition metals into those carbonates, which can differentiate those two minerals. Thanks.
When we did elemental analysis, we saw, and also XRD, they were both calcium carbonate. One was just aragonite and one was calcite. I missed the first question though.
Oh, it's about the difference of the hardness of the minerals. Why is that?
Oh, the hardness. I think that's just because one is aragonite and one is calcite, but also because, as you saw in the SEM scans, oh, it won't let me go back now in the presentation. That's odd. Okay, there we go. Okay, so also what I was thinking is that with the calcite, there's a lot of this really soft and not super, like all this air inside, and it's not very strong yet, this layer right here. So I think that whenever I went with a drill, I just kind of destroyed a lot of this layer, versus here I also went with the aragonite and the acicular sample, I kind of, instead of going inside, because these scans were taken sort of inside of the needles, here I went just on like the top, and it was very solid. So if I show you the CT scans, now I have to go really back, sorry. So this is pretty solid. So when I came with a drill, it was a lot harder than this one that was hollow and not as fused together as this one is. The scans that I took of the acicular are only like on the edges and inside of little cracks formed by the different needles. And with the tubular, I kind of went with a scalpel and dug a little hole so that I could actually see inside. So that also, that shows the difference in the scans. Does that answer your question?
Yeah. The thing the reason why I'm curious about this is because I saw some few papers about the incorporation of organics, for example, some small molecules into the calcite structure. I was wondering, maybe microbes can incorporate some organics, small molecules, so that it can change the physical properties of the carbonate. But that's just my guess. Yeah,
that's the research pretty much, is that microbes inhabit this morphology, which is abiotic, and then somehow create this morphology. So yeah, it's interesting.
Okay, thanks.
Yeah.
Excellent. Are there any more questions at this point?
One.
Okay.
This is David. Is it feasible to, or did you try to grow whatever is growing in that petri dish on a single crystal of material as a substrate?
That would be awesome. Yeah, I mean, to prove this formation, we would want to be able to grow. First we would have the abiotic, and then we would try to grow off of that, have microbes grow this one. I know that Nicola did that analysis with the agar plates, but yeah, it does need to be lab tested, if that was your question.
It just seems like if you have a single crystal of aragonite and you put some agar with your bacteria on it, and sit it there for a few days, and then go look at it under SEM, you might see whatever the critter is gnawing away into the crystal.
Yeah, that would be really awesome. That is kind of our plan, to kind of go back and get better samples and do the biotic analysis. We would need to get the critters. So yeah.
So has someone at all tried to analog grow these kind of structures in general in a lab?
These specific ones, no, but the process isn't completely unheard of, but it is new and still there's not a lot of information on it yet.
There is a very old paper from the 40s, right? Which also has biased a lot all the field, speaking with a big guru of life in caves like Penny Boston and so on, according to them. Anyway, such paper shows that you can grow, for example, the acicula, right? The acicula can be grown in the lab, and there is a model for explaining such morphology.
I mean, acicular growth is just pretty standard aragonitic growth in supersaturated material.
Yeah, exactly.
So I would definitely look into the geobiology literature for that kind of thing, because that's something that a lot of people do with like hot springs precipitates or marine precipitates.
Yep.
So
absolutely.
You may not have to do it yourself if you know that you've got the same kinds of bacteria. Or if you've got something new, that could be a whole new interesting experiment.
Right. So I would like to chime in also for about David's question. I think it's a very good idea. I think it should be done. However, it's not probably very simple. We try while at ETH and we study the asparage, we try to grow exactly those bacteria and put in some aragonite, some calcite, and some heavy metal substrate, and to see where the bacteria would have, grow towards. And the agar plate have been totally overwhelmed by bacteria that are normally in this environment, especially molds and molds, especially molds. So the impression is that niche, right, inside those caves, they have something special. Like the first question that Connie asked here, right? Why only in that area of the cave? And we don't know yet why. It's a key question, I guess, right? So if you bring those bacteria outside, they might be they might not be in their natural environment. Therefore, they might lose against many other form of life.
Excellent. I have one more question, but are there any other people who would like to ask a question?
Okay, one more. You didn't say, as far as I heard, anything about the rate of growth of these things.
No. Correct me if I'm wrong, Nicola, again, but you would have to go back and often visit and take measurements and such like that. And Nicola visited this particular cave once, and we haven't grown these, at least from Breezeway, so we don't know the growth rate.
So, are these
1,000 years old or 1,000,000? That's the question. Or more or less?
It has to be younger than the cave. So there's that.
That's a good argument.
Thank you. So, but other than that, no, I'm not sure if we can date them. I'm not sure if we could, because they're just calcium carbonate, but I might be wrong about that.
Might be able to do like carbon or uranium thorium if you have enough material.
Yeah.
Okay.
So if you go back to respond to them, because this is again right, studying these things open up more questions than what he gave responses to, right? If you go back to the slide 16, please. Just want to show you that those calcite structures that we grew in the agar plates, they grew in roughly a couple of weeks. And so let's go back.
Sorry, he's not reacting to me. There we go.
There we go. Yeah. So those structures there, they were, you know, roughly 100 microns in diameter, which if you go, so yeah, there are those black dots inside the agar, right? Those one formed in roughly a couple of weeks, and they were colonized by life because they're full of holes from where you can see stems coming out, right? So they're clearly their formation is clearly controlled by life. It's not that it's simply a abiotic precipitation of nodules of calcite, right? I don't have the
And if you extrapolate that growth rate and the volumes, can you come up with a number that is even a first order? Yes?
Yeah, you could try to do that. However, the agar plate is a super rich, you know,
I wouldn't give you an accurate reading.
It would be like a
agar plate is like a bunch of Americans in a supermarket full of toilet paper when there is a pandemic, right?
I thought it was Australians with toilet paper, but you know.
Right. So their activity is boosted, right, by the presence of the peptone and all those things that you put inside the agar plate to boost them up, right? Yeah.
But uranium, thorium, that hasn't been tried yet.
Yeah, might work. But yeah, I mean, try to extrapolate from the agar plate. It should be done. Maybe it will give us. Maybe, you know, maybe will give us rates of growth that will be so high that are impossible, because cavers have been in Asparage for 30 years now, and I don't think they have noticed the branches becoming double size, right? There are pictures, right, from, yeah.
You could also spike one of the growing things and then sample it in 10 years and see. But
Yep.
Directly in the cave, you mean?
yeah,
Yeah. They don't like
people do growth rate studies on speleothems all the time. I'm sure someone in Jay Banner's group or Dan Breaker's group has done something.
Yeah, they do. They measure in inner space caverns. I know they do there.
Okay, so I have one more question. So you obviously talked about that you're also curious to find these kind of structures fossilized, right? Or in the sense, maybe on a different planet or something. So what are like the, I don't know if you know that, have these structures been found like fossilized, like in the sense of millions and millions of years old, or what are the oldest, fossilized is the wrong word, but what are like the oldest structures that you have been found who look somewhat like that?
This goes back to the question of how old are they? We don't know. We just know that if they were undisturbed and also the cave didn't collapse or something like that, then they would be, once they go from acicular to tubular, then, and they stay that way, then we can say that if undisturbed, we could say, oh, okay, we see this morphology. At some point, there must have been life. But of course, with caves and all that, I mean, they could be collapsed, destroyed, anything like that. But it's just a potential way you could search for life.
But I guess
Microproblematica.
Have you found, has any fossil morphologies like this been found in the record anywhere on Earth?
Not that I know, but Nicola?
So there is a cave nearby Breezeway, which has similar shapes inside the sediments in the cave themselves, which are very similar to this concretion. So but also that cave is not accessible. But there is an article from Davis that shows these structures. But that's very recent sediments, right? Probably 10 or 20,000 years ago. Not in the old geological record, now that we know, not to our best knowledge. But we should probably talk more with the paleontologists, right? Because
there's a whole subcategory of microproblematica that are, I mean, not so much in the terrestrial realm that I'm aware of, although there certainly may be, but like there's a bunch of stuff that's just like a circle of acicular cemented stuff that doesn't have a particular form, but always forms in the same way. So we're going to give it a name. So, I mean, you know, it wouldn't surprise me.
Yeah, something similar that I have been thinking about, but maybe it's worth comparing. There's a late Triassic impact crater called Rochechouart in France, which had a hydrothermal system. And in these rocks, we find some of these like tubes as like the broadest, you know, like very, like obviously a microfossil kind of tube thing, which has been hypothesized to form like with biotic help. But yeah, as I said, I'm defining it so vaguely because it's not really well explained. So yeah.
There are literally microproblematica called microtubus.
Yeah.
Which is just a small calcareous tube that we don't know what it is.
Exactly. So when I saw your talk, I thought that was kind of interesting. So I should dig up a paper and think about and talk to you guys how that might compare.
Yeah.
Field trip to Rochechouart.
Yeah.
I think it could be a good pitch for the Habitability Center, because understanding all the fossils that we see in this planet might help understanding the fossils that we might see in other planets, right?
Isn't that the whole point of like Habitability Part 1?
Yeah, exactly. Well, nice. Yeah. So we should for sure speak more with paleontologists and with Connie. Yeah, and Roland. Yeah.
Super cool stuff though, Carole.
Very cool.
Yes, very cool. Yeah, that was a lot of fun.
I'm glad.
Thank you.
Anyone else with the last? We have even a little bit of time left, so if there are any more questions, that would be a chance. Okay, it looks like we all talked out, but that's okay. We talked a lot. It's excellent. Okay, thank you so much for giving this talk today to both of you. Yes, and I think we all hope to hear more after we had so many questions. So if there's an update, obviously contact us.
Great, thank you.
We will keep in touch, and thank you, Carole, for presenting.